The purpose of this video is to review over the cellular structures found in a common eukaryotic cell, specifically the structure and function of the basic organelles found in the cell. Now remember that the study of the cell is known as cytology, and that the cell is the smallest unit that can carry on all of life's processes. The cell was first seen in 1665 by Robert Hooke. When Hooke was examining the cork wood, he noticed that the wood was divided into little squares or cells, and these cells reminded him of the rooms of a monastery, and the rooms of a monastery were known as cells, and thus Hooke is credited with naming the cell. The first living cell was identified by Antoine von Leeuwenhoek in 1675, when Leeuwenhoek was identifying little tiny one-celled organisms in pond water. He thought that these organisms looked like little tiny animals, and he called them animalculae. One of the fundamental theories associated with the cell is the cell theory. The cell theory has three components to it. The cell is the basic unit of life. This was presented by Robert Hooke. All living things are made up of one or more cells. This was presented by Slindler and Swan, Slindler being a botanist studying plants, Swan being a zoologist studying animals. Both stated that all living things were made up of cells. And then finally, all cells divide and come from old cells, which is from Virchow. Now, when we look at the cell, we can divide the cell into two basic types. Eukaryotic cells, which are organisms that have a membrane-bound nucleus, and prokaryotic cells, which are organisms without a membrane-bound nucleus. The prokaryotic, pro means before, and it is an evolutionary reference by evolutionists who believe that the prokaryotic cells evolved before other cells. The prokaryotic cells are found in the kingdoms Eubacteria and Archaebacteria, while the eukaryotic cells are found in the kingdoms Animae, Plantae, the fungus, and Protista. Now this video will be specifically covering the eukaryotic cell and the structures associated with the eukaryotic cell. Eukaryotic cells, again, are organisms that have a membrane-bound nucleus. They have DNA. Eukaryotic cells can be either unicellular or multicellular, but all will have organelles, and each of these will be surrounded by a plasma membrane. This plasma membrane will surround and protect the cell. Additionally, a nucleus will be found, which is the largest organelle. It contains most of the genetic information. And then again, the organelles will perform specific functions. The functions will be like little tiny organs, and they will be analogous to organs of a multicellular body. Now, as we begin, not all eukaryotic cells have a cell wall. However, plant cells do. The cell wall will protect the cell. It gives the cell its shape. It's made up of cellulose and will be found specifically in plant cells, algae cells, fungi cells, and in most bacteria cells. We will begin with the cell membrane in describing the structures associated with a typical cell. The cell membrane is also known as the plasma membrane. It is the outer covering, that the protective layer around all of the cells. For cells with a cell wall, the cell membrane is inside of the cell wall. Now, the cell membrane allows food and oxygen and water into the cell and waste products to move outside of the cell. This cell membrane is found on all cells. It is the boundary of the cell separating the inside from the outside. It is known to be semi-permeable. It has a semi-permeable membrane, meaning that it allows some substances into the cell while it keeps other substances out of the cell. The cell membrane has a phospholipid bilayer. This phospholipid bilayer means that the lipid molecules are fluid and can move past one another in a fluid manner. It also allows for protein to move and change in this layer. So scientists have explained the cell membrane using a model known as the fluid mosaic model. It is fluid because individual phospholipids and proteins can move around freely within the layers like it's a liquid. Mosaic because of the pattern produced by the scattered protein molecules when the membrane is viewed from above. So the phospholipid bilayer is the basic structure of the cell membrane. It has two layers of phospholipids with a hydrophobic tail protected inside by a hydrophilic head. The hydrophobic tail is water-fearing and not attracted to water, so it is nonpolar. The hydrophilic head is attracted to water and thus is polar. The average thickness of the cell membrane is about 7 nanometers, and the fatty acid tail of the phospholipid can be saturated, which is straight, or unsaturated, which is bent. Proteins can also float or be fixed and also have hydrophobic and hydrophilic portions, just like our phospholipid bilayer. So in summary, the phospholipid bilayer is semi-permeable and it acts as a barrier where it will allow some material to pass through the membrane while other material it will not allow to pass through. It acts as a barrier to most water-soluble substances due to that hydrophobic tail region. The gelatin-like material inside of the cell membrane is known as the cytoplasm. All cells have cytoplasm. The cytoplasm is constantly flowing. It is also known as the protoplasm, and it contains the various organelles of the cell that we will be discussing. Now, as we begin to look at these internal organelles, remember that each organelle will have a specific structure and function, and the function will be analogous to an organ of a multicellular body. So we will see a correlation between the structure of the organelle and the function that they perform. Many of these structures will be key as far as identifying an organelle. They will have a very distinct appearance, and so you do need to be aware of not only their function but their descriptive analysis as well. The largest organelle of the cell is the nucleus. The nucleus is the control center of the cell. It directs all of the cell activities. It contains the instruction for everything that the cell does. This means that the DNA or the genetic material is found within the nucleus. Here we have a photograph of an actual nucleus. A few things that I want to point out to you is the outer edge of the nucleus is known as the nuclear envelope. We have the nucleoplasm inside of the nucleus, and then we have this structure right here known as the nucleolus found within the cell. Cells can have one or more nucleolus present, and the nucleolus will help with ribosome assembly. So here we have the nucleolus. Nucleolus means little nucleus. It is found within the nucleus of the cell. It contains the RNA and proteins for ribosomal synthesis. Also notice that you will see the term chromatin. And I do want to stop and describe the difference between the term chromatin, chromosome, and chromatid. Chromatin is unwound chromosomes, and you will see this within the nucleus of the cell at various stages of cell division. Now here we have an intact chromosome. Now the chromosome is made up of two parts. Right here we have a chromatid, right here we have a chromatid, and right here in the center where they are attached is the centromere. So a chromosome is composed of two chromatids attached in the center by a centromere. Chromatin is unwound chromosomes. Now the chromatin contains the genetic codes that will control the cell. The chromatin is made up of DNA and protein. And so you will see DNA and protein in both chromatin and in chromosomes. Centrioles are organelles that are described as short cylinders. They are typically found near the nuclear envelope that we saw a couple of slides back. They are generally at right angles to one another. There are two that are present, and they help control cell division during mitosis. And specifically, they help the chromosomes to move and line up along the equatorial plane during metaphase of mitosis. The endoplasmic reticulum is abbreviated ER many times, and it is a series of folded membranes that move material, specifically proteins, around in the cell like a conveyor belt. The endoplasmic reticulum is divided into two types. The smooth endoplasmic reticulum, which does not have ribosomes present. This functions in lipid synthesis. The rough endoplasmic reticulum has these little dots that you see, and those are ribosomes that are present, and this functions in producing proteins. These ribosomes that we just saw on that previous slide help to make proteins. We can find these ribosomes floating free within the cytoplasm or attached to the endoplasmic reticulum, specifically to the rough endoplasmic reticulum. The ribosomes are made within the nucleolus and are small particles of RNA needed in protein synthesis. The Golgi body, Golgi complex, or Golgi are described as a series of stacked flattened membranes. They sort and package proteins, and within plant cells they are called dictosomes. Dictyosomes. Again, they are called Golgi bodies in the animal cell and dictosomes in the plant cell. The organelle of lysosomes. First off, the word lysosome is Latin for kill body, and the purpose of the lysosome is to digest things. They might be used to digest food or to break down cells when it dies. They can break down food particles, cell waste, or worn out cell parts. The mitochondria is known as the powerhouse of the cell because this is where ATP energy is made in the process of cell respiration. So this is the organelle that releases energy from the food, and this energy is going to be broken down into carbon dioxide, oxygen, and then ATP energy. Here we have an electron photograph of the mitochondria. The folds of the mitochondria are called the cristae. This is where the electron transport part of cell respiration occurs. This open area of the mitochondria is known as the matrix, and this is where the Krebs cycle occurs. Outside of the mitochondria in this area is the cytoplasm or cytosol, and this is where glycolysis occurs. So we will be discussing the mitochondria in further detail on my videos covering cell respiration. The organelle that functions for temporary storage space is known as the vacuoles. The vacuoles can be found in all types of cells. In plant cells and in protein cells, we may see these as storage for water, especially plant cells, We may see them as food storage, as in a paramecium or an amoeba. We can see them as waste storage in various cells. In animal cells, the vacuoles are very small, and we will see the vacuoles associated with the movement of material into or out of the cell by the processes of endocytosis and exocytosis. We will be discussing the movement of material into and out of the cell on part 2 of this video. The cytoskeleton is the scaffolding-like structures in the cytoplasm that helps the cell maintain or change its shape. It is made up of protein, microfilaments, and microtubules. The proteins that are associated with the cytoskeleton are the proteins actin and myosin, which help the cell to change its shape. The microtubules that are found with the cytoskeleton all help the eukaryotic cells function in cell support. The microfilaments are built from actin, which is a globular protein, and it functions in support of the cytoskeleton and for localized contraction of the cell. Additionally, some cells may have intermediate filaments. Now, the size of the intermediate filaments, they are intermediate to microtubules and microfilaments, and they function in reinforcing the cell shape. The short hair-like projections found on cells are known as cilia. Cilia are found on a wide variety of cells. In paramecium, they cover the outer part of the cell on all surfaces. In humans, we will see the cilia associated with the throat and moving material out of the throat when one coughs. So they would be found in the trachea area associated with tissue known as pseudostratified ciliated columnar epithelium. Now, this is the area of the trachea when one wakes up and coughs up mucus. This is the tissue that has the cells that helps move that mucus up the trachea so that one can cough it up. The long whip-like organelle that functions for movement is known as the flagella. We will see flagella on organelles such as trypanosomes or euglenas. In the human body, the only cell that has a flagella are the sperm cells. Additionally, extracellular material can be associated with the cell. This is material that's found outside of the cell. It can be secretions by the cell, such as saliva or gastric juices, etc. And we'll discuss more of this on a future video.